Okay, thanks, Zach. And thanks everyone for joining today for our last webinar. And yeah, as Zach mentioned, I'll be going over the PEAKS Glycan Module within PEAKS 11, which is new to our PEAKS Studio software. So here's the webinar agenda. So I'll start out by giving an overview very briefly about protein glycosylation, background on glycoproteomics, and a little bit about how the Glycan module works in terms of the software workflow. Next, I'll show how Glycan module can differentiate between isomeric glycopeptides. Then I'll go into a sort of case study, so reanalyzing some previously published data to show how Glycan module can identify optimal dissociation methods for glycoproteomics. Then I'll talk about the importance of A-score and how A-score can assign some confidence in the glycan or the glycosylation site within the glycopeptide. And then lastly will be another case study showing how we, in our lab, identified some differences between the glycoforms of a recombinant protein that was expressed in two different host cell systems. Okay, so just to start off, brief introduction on protein glycosylation, which many of you probably already know, but a range of different glycosylation types exist across eukaryotic cells and mammalian cells. And the cells have multiple different pathways for protein glycosylation. And it often involves the secretory pathways comprising the ER and Golgi apparatus. And so the installation of glycans on proteins with end-link pathways end-link pathways target the nitrogen atoms of asparagine residues, whereas O-link pathways will target the oxygen atoms of serine and threonine residues. And we also have C-link pathways that can target the carbon atoms of tryptophan residues. And many of these glycosylation events are observed on proteins known to be secreted or displayed extracellularly, owing to their role of glycosylation in mediating extracellular protein stability and function. And we also have membrane-bound glycoproteins. Intracellularly, O-link glycosylation also occurs and has a crucial role in cell signaling events. Sorry. So there's a range of common glycan classes that are observed across mammals, but mainly end-linked and mucin-type O-link glycosylation can occur. So end-linked glycans include polyamminose, oligomannose, and complex and hybrid structures. And this polyamminose carries 1 to 3 mannose residues with variable core fucosylation events. And oligomannose glycans contain terminal branches composed of mannose sugars. Complex and hybrid glycans may contain galactose, glycnac residues, as well as galanac and fucose, and other monomers such as N-acetyl-neurominic acid and glycol-neurominic acid located on their antenna. And so glycoproteomics aims to identify the identity and the position of glycans on proteins across an entire proteome of a given cell or tissue. And this is achieved mainly by LC-MS analysis, which has enabled this large-scale global analysis of glycoproteins. And it's the structural diversity of glycans and heterogeneity of glycosylation sites, especially with O-link glycans, that presents a significant challenge in terms of a comprehensive analysis and identifying all the glycan identities and glycosylation sites across a proteome. And so in order to kind of overcome this challenge, we need software to do this. And so that's why we developed PEAKS Glycan Module. As Zach mentioned, it's a glycopeptide-based approach for both N-linked and O-linked glycosylation. It assigns glycans sites specifically within a peptide, and we provide a score that can provide the user some confidence in whether the glycan is accurately localized to a specific site within the peptide. With Glycan Module, we can do glycan profiling, so looking at differences in abundance of glycans at a particular residue across multiple samples. And then we do accurate glycopeptide detection. If you have ion mobility data, we can do 4D precursor feature detection. We support common glycopeptide fragmentation and instrument methods. And we also provide this S-score, which can be used to annotate a structure for glycan composition. So I'll discuss these points a little later in the talk. So here's our Glycan Module workflow. So starting with raw LC-MS data uploaded into PEAKS 11 Glycan Module, the data first undergoes data refinement and then passes through this spectrum classifier where we can extract from the MS1 scans, we extract the features that will be used for quantification analysis. And we also classify the spectra, the MS2 spectra, as either non-glyco MS2s or scans that can be predicted to contain glycan fragmentation ions. And this is mainly based on the fact that for peptide fragmentation ions or MS2 scans, we see most fragment ions in the lower M/Z range, whereas with glycosylated peptides, we often see higher M/Z mass fragment ions. And so from the non-glyco MS2 PSMs, we perform, or scans, we perform a database search by searching a protein sequence database that the user has put in. And then from here, we can identify potential protein candidates. And this protein candidate list is fed into the Glycan Module search, which begins with the glyco MS2 scans. And so in addition to the protein sequence database, we provide software a glycan database so we can match the fragment ions to predicted or expected fragment ions within the glycan database, as well as assigning it to a peptide from our protein candidate list. And combined, we come up with a list of glycopeptides and glycoproteins that's in the final result. And so all of these are filtered or controlled by FDR, as well as both peptide and glycan scores. You can set a specific score threshold for each of those. And in addition, we have a partials tab, which I'll show later in the software demo, where if we only have a glycan ID from a particular scan, we can, and say we don't have good fragmentation ions supporting a peptide sequence, we include these in the partial scan. And as well, we can have a good peptide sequence without a confident or a glycan match. And we'll assign a mass tag to that peptide sequence and include those in the results. So PEAKS Glycan Module supports various different instrumentation types. So we can do Orbitrap and HCD, as well as EThcD, which has become a more popular method since the EThcD allows us to observe more intact glycan fragments. We could also look at timsTOF CID data, and also the instrumentation method that's becoming more popular for glycoproteomics is xenotOF EAD, because again, we retain a lot of these intact glycan fragments associated with peptide fragments. And as well, we have higher intensity fragment ions that can provide us the peptide, or derived from the peptide backbone fragments that can provide us the peptide sequence. So now I'll show you how PEAKS Glycan Module can differentiate between isomeric glycopeptides. So there have been many studies recently published where they've analyzed or characterized IgG proteins. And when you pull down IgGs or antibodies, say with protein A or protein G, you often get a mixture of different IgG subclasses, mainly IgG 1 through 4. And within the heavy chain constant region, there's this conserved glycosylation site. And often from tryptic digestion, you get these isomeric peptides being produced, where at these positions indicated here, you can have either tyrosine or phenylalanine or a combination of both. And in terms of IgG3 and IgG4, these peptides would be isomers of one another. And so we need to be able to accurately map these to the correct IgG subclass in order to characterize their glycan profiles. So you can see we were able to do this here. Our spectra that mapped that glycosylation site. The bottom two here are basically the same glycan from IgG3 and or same glycopeptide from IgG3 and IgG4. They're isomers, but just having the tyrosine and phenylalanine swap between the two positions, we're still able to map them to the correct IgG subclass. And here's what it looks like from a protein coverage view. So here's the coverage at that site for IgG1. You can see extensive glycosylation at that site. This is IgG2. And then the two isomers that are hard to differentiate, IgG3 is shown here, and then IgG4. So just highlighting the fact that within a mixture of IgG subclasses within the same sample, we can still accurately map glycopeptides that are very similar to different IgG subclasses. Okay, next I'll show kind of a reanalysis of previously published work where they've identified optimal dissociation methods for glycoproteomics. So this work was published from Carolyn Bertozzi's group in collaboration with Nick Riley, Stacy Melliker, and Mark Dreisen. In 2020, they published this analysis where they systematically assessed kind of different dissociation methods, starting from EThcD and then EThcD supplemented with HCD, so EThcD scans, and also assessed step collision energy, HCD. And in terms of glycoproteomic analysis, they had a number of different criteria that they kind of scored the different methods on against. One was acquisition speed, peptide fragmentation, glycan fragmentation. So they talk a lot about sequence coverage for both peptides and glycans from the different methods, as well as the ability to localize the glycan to a specific site within a glycopeptide, and differentiating between the best method for either N-linked or O-linked glycopeptides. So these are the different dissociation methods that were tested in the paper. So there's 14 total here. And this is just one of the figures taken from their paper looking at N-glyco-PSMs and the number that can be identified by each dissociation method. So you can see that EThcD is sort of the lower end of the scale in terms of IDs, whereas HCD and step collision energy HCD achieve the highest number of glyco-PSMs. However, when you look at the peptide sequence coverage and glycan sequence coverage, and this is the percentage of possible fragment ions, or in terms of peptide sequence coverage, it's the possible number of peptide backbone fragments for that peptide and the ones that were observed. And same with glycan fragmentation ions. They look at the number of possible glycosidic bonds that could be fragmented, and this percentage represents the number that were observed. And so for EThcD, it actually outperforms HCD for the most part in being able to cover most of the peptide sequence and glycan sequences, which helps in terms of localizing the glycan to the correct glycosylation site. So all this work was done in Bionic software. So in addition to assessing different dissociation methods, I think it's important to assess how different types of software result in glycoproteomic analyses, and whether you see the same trends across different software platforms. So I ran this in PEAKS Glycan Module, and so this is first showing the analysis of, they have this standard glycoprotein mix of 9 different glycoprotein standards, and then performing each run with the different dissociation method. So overall, we see fairly consistent coverage across all of the different methods, a slight drop in protein coverage when using EThcD, which may not be too surprising, since without having the HCD supplementation, we don't see good peptide backbone fragmentation. But overall, fairly reasonable protein sequence coverage across all of the methods. So next I looked at the overall total number of glycopeptides that could be identified from each method. And, oh, sorry, this is just what the protein coverage looks like, which I'll go into a little later in the software demo. So in terms of N-linked glycopeptides, it appeared that HCD methods far outperformed EThcD methods in terms of total numbers, whereas HCD with 30% collision energy achieved the highest number of N-linked glycopeptides, and HCD of 25% collision energy achieved the highest for O-linked, again, far outperforming the total number of EThcD glycopeptides identified. And these are all peptides that passed our FDR cutoff. And then when looking at the step collision energy, we saw a further increase in the number of glycopeptides that could be identified. So it seems like having a combination of 3 different kind of stepped energies provides the best identification results for glycopeptides for both N-linked and O-linked. So that all sounds good, but it's more important that we have accurate localization of glycans within our glycopeptide IDs rather than just reporting the total numbers identified. So next, I looked at peptides that scored above our A-score threshold, and those that we can confidently assign a glycosylation site to. And so this is showing the percentage of glycopeptides where the glycans had an A-score above 20. And so for our A-score, an A-score of 20 represents a probability value. And so it takes into account the other possible modification sites within a peptide. And so an A-score of 20 is equivalent to a p-value of 0.01, and then the maximum A-score is 1,000, which represents or indicates that there's no other possible sites where that glycan can modify an amino acid within the peptide. And so overall, the step collision energy, HCD, improved glycopeptide identification without really sacrificing glycan localization. So here's the percentage of peptides that pass the 20% or a score of 20 out of the total number of glycopeptides identified. And we see kind of similar values with an A score of 1,000. However, you can see that EThcD overall had a higher percentage of glycopeptides where we can confidently map the modification sites. And another way to look at this data is if you want to compare the glycans that are identified across the different dissociation methods, we have the glycan profiling tool where you can select either different glycans as well as a specific residue or glycosylation site within a protein, and just see whether there are differences between the different dissociation methods in terms of detecting different types of glycans at that site. So for the most part, what I was seeing was that if the glycan is fairly abundant, there's really no issue in detection. Most methods will detect it if it's an abundant glycopeptide. For the lower abundance ones, there is quite a bit of variability where certain methods perform better than others in terms of detecting those glycans. And here's just another site from the same protein, where again, we see a similar trend, and actually some other glycans that are fairly more abundant, we're readily detecting them with certain step collision energy methods and not others. So it really depends on the glycans that are present. Maybe it's related to the glycosidic bonds that need to be broken in order to identify these glycans and glycopeptides with respect to the right dissociation method. Okay, so next I'll go into A score and how we assign confidence in glycan localization. So there are kind of three main ways, and they discuss this in the paper that I was just mentioning. Three main ways of localizing glycans within a particular glycopeptide. One is you can have an intact fragment, and by that I mean you have a peptide backbone fragments that retain the intact glycan moiety. So you can assign it to a specific site when the glycan's intact and that you have a glycan fragment. The other types of ions within a spectra that can be used are hexnac retaining fragments. So you have peptide backbone fragments that may lose most of the glycan moiety but retain just the hexnac monomer. And also, mostly in the case of end-linked glycans, if there's only one possible glyco site with the end-linked motif present, then it becomes a bit obvious that that's the only possibility. So you can assign the site there. So here are some examples of that. Here we have just one possible NXT and glycosylation motif present in the peptide. So in this case, again, it becomes a bit obvious, but we also have the full intact glycan associated with the peptide ion. And so in this case, we assign this an A score of the maximum of 1,000. And here's an example of a peptide fragmentation ion that's still associated with the full intact glycan. Okay, so here I want to highlight the advantages of EThcD over HCD methods in glycan localization. So here's the same glycopeptide from the same sample, just run in HCD versus EThcD mode. So here you can see we really don't have a lot of intact glycan fragments still present in the spectra when using HCD dissociation. We do have some smaller oxonium ion products present, but really no fragmentation ions that would allow us to localize this glycan to any particular serine or threonine residue within the peptide. So we're really not confident where this glycan is attached. But as you can see with EThcD, we have a lot more intact glycan fragments. And in addition to B and Y ions, we also have C and Z ions that can be used in order to provide some confidence in the localization site, especially when there's kind of an enrichment of serine and threonine within the peptide. Here's a more complicated example where, but it's fairly common that you can see more than one O-linked glycan attached to the same peptide. And so it's really important to have a number of different fragment ions still associated with intact glycans in order to accurately map these to a specific serine or threonine within this peptide. And so I've highlighted within Glycan Module under the spectra, we provide the glycan ion match table. And I've highlighted the ones here that are important for assigning a localization. And so we have, we have B and C ions along the bottom here, and Y and Z ions. So where I've starred them, we have fragmentation ions with some glycan fragments or intact glycans associated with each, supporting the localization at threonine at position 4 and 8. You can see we're still not entirely confident in the localization. There are some other possibilities still, but based on the fragment ions we see, we can provide some confidence scores here. Okay, so lastly, I'll show a case study that was performed in our lab where we characterized differences in glycosylation of a recombinant protein expressed in different host cell systems. So this protein CD33 was purchased from R&D Systems and SinoBiologic. And so the protein prep from SinoBiologic was done in HEC293 cells. And from R&D Systems, they expressed it in CHO. So we started with 5 micrograms of each. We did our reduction alkylation procedure and digested it with trypsin and ran it on our timsTOF Pro. And then in Glycan Module, we used the following parameters. And then any peptides passing these filters, we compared the glycan profiles across the 2 proteins from different expression systems. So overall, the coverage looked fairly similar. All the blue bars represent non-glycopeptides. The purple are end-linked glycopeptides. So we had really good coverage of 6 different end-linked glycosites across these proteins. And we also saw some truncation of the N and C terminus, probably a result from different expression vectors and sample prep. So when we compared the glycans identified from each protein, we saw significant differences. And yeah, roughly, only half of the glycans overlapped between the 2 proteins, even though they're the exact same sequence, but different host cell systems will result in different glycosylation events. And I think this is important to consider when purchasing recombinant proteins from a supplier, as glycans and different glycan and glycoforms can alter structure and function of proteins. And one way to look at the differences is by using this glycan site profiling tool within Glycan Module, where you can click on a specific residue. In this case, it's asparagine at 209 and CD33. And looking at this pie chart, you can see there's significant differences between the 2 expression systems. And mainly in CHO cells, we see a significant increase in fucosylated glycans present on CD33. Okay, so with that, just some conclusions. So isomeric glycopeptides can be differentiated in glycan module. EThcD results in the best glycan localization, while step collision energy HCD overall identified the most glycopeptides from the glycoprotein standard mix that was analyzed. A-score can provide users some confidence in assigning the correct glycosylation site within a peptide. And the glycan profiling tool can be used to compare different glycans, identify specific sites that result from differences in dissociation methods or host cell expression systems. And in the CD33 case study, only 48% of the glycans were shared. And in CHO, we saw a higher level of fucosylated glycans. Okay, so I'm just going to switch over to the software now and just demonstrate a few different features of Glycan Module. Okay, so here we have the project which I ran with CD33. And I just wanted to highlight how the setup works. So within PEAKS 11, we go back here, you would, after you upload your raw data files and set your instrument type and so on, you would then go to the workflow selection. And instead of doing a DB or PEAKS-Q, you'd select this glycan search workflow. You can choose whether to associate feature with Chimera scan. And then the rest, this part will look the exact same as a typical database search, but below we have this glycan option. So you can select whether you want to do N-linked or O-linked analysis. If you have samples where you've enriched for glycopeptides, you can select this and search only for glycopeptides if you'd like, and choose whether to select certain adducts per peptide and also maximum number of fucose monomers per glycan. You can also choose whether you're doing composition analysis or structure analysis. So for every glycan composition, there could be more than one structure. And because we use a linkage-based glycan database, we can extract some structure information from it and potentially assign more than one structure for a particular glycan composition. So that is an option, or you can just set as composition, and we'll only provide the one glycan composition per glycopeptide. We also have a number of report filters, so you can filter the peptides and the glycans by a specific score, or you can choose to filter them by FDR. It helps to save your workflow and give it a name. So when I was running the samples for different dissociation methods, it helps to save, say, an HCD workflow so that when you're running multiple different analyses, you ensure that all the parameters are the exact same. Okay, so this is the protein coverage view. As I mentioned, any of the glycosylation sites you can select to bring up this pie chart and view kind of the relative abundances of each glycan at that site. And also, any of these glycopeptides can be selected to view the MS2 spectra. We also have the glycopeptide tab, so it lists all of the glycopeptides that passed your score filters. And all of these are interactive, so you can go in and zoom in and zoom out to view the MS2 or the fragmentation ions. And the one thing I wanted to highlight is the partial tab, which I mentioned earlier. So even if we don't have a good glycan ID or peptide ID, we'll still list them in this partial table, which can be helpful if, say, you're expecting a certain glycan, but maybe it wasn't in the glycopeptide result list. And so here's an example of where we've identified a peptide and matches to the database and has a good score. And we have these fragmentation ions that are in the higher M/Z range, but we haven't been able to assign them to any particular glycan. There could be many reasons for this, including it might be a novel glycan that's not present in our database. So we'll just assign a mass tag to this peptide and include it in this list. And also, we can have glycopeptides where we have a glycan and good glycan fragmentation ions to support this ID, but we just haven't been able to sequence the peptide. So we'll assign this mass tag associated with this glycan moiety there. Sorry, I can't quite see this. And lastly, I'll just show you the glycan profiling tool. So here's the step collision energy dissociation methods that I've ran from the glycoprotein standard mix. And so there's this tool in the protein coverage view called glycan profile. And if you click that, you can compare the different glycans, in this case, the different glycans that were identified across different methods. And so we have options to select specific glycans. So if you want to just look at the N-linked ones, you can select those from the list. Or if you're interested only specific glycan moieties, you can select them there, or specific samples. And so shown here at position 99 within this particular glycoprotein, we can view the differences in glycan IDs at that site across the different step collision energy methods. And here it's showing as a percentage of glycans at that site. You can switch to showing the area under the curve values from the precursor profiles for each glycopeptide. Okay, so that's basically everything I wanted to show in terms of glycan module in the presentation. But yeah, I've been happy to take any questions.